Been looking at biomolecules, and for biomolecules, you need to understand carbohydrates, proteins, lipids, enzymes, nucleic acids. This is the first part of biology 1400 and it's called 1401. Second part will just be molecular biology and genetics, which is just two topics, that is biology 1412. But 1401 and 1412 are combined and they'll give you 1400. So we are now looking at lipids. Since we've done carbohydrates already, we've done enzymes, we've done proteins. Okay. So first thing first, remember what is a lipid. When you talk about a lipid, it's basically a group of these are naturally occurring esters. And I think from our grade 12 we know what esters are. But one important thing is that these are naturally occurring esters. We don't make them like that. You just find them in nature. Okay, that's how they are. Esters are formed from two functional groups. What functional groups are those? They are formed from this functional group. This is a carboxylic acid functional group or a carboxyl and an alcohol, which is OH. These are the two functional groups which will form an ester. But to be specific for lipids, this carboxylic functional group is found in a molecule known as a fatty acid. And then the OH is going to be in alcohols. So now you can define well what lipids are. Lipids are naturally occurring esters of fatty acids and alcohols. Remember, an ester is a compound formed when the carboxylic acid functional group, which is carbon, double bonded to oxygen, then bonded to OH, gets to connect with a compound that contains OH as a functional group. So this reaction is always a dehydration reaction because water is going to be released when these two combine. We have got different lipids that we are going to study in this session, and this is going to include number one, fats. Okay, so supposed to understand fats, you're supposed to understand waxes, supposed to understand also you have got vitamins which you call fatty soluble vitamins. These are also lipids. What are for fatty soluble vitamins? These are vitamins including we can abbreviate them as ADEK, vitamin A, D, E, and K. These are fatty soluble vitamins. Apart from that, we also have what we call glycerides. These are also lipids. And phospholipids. So by the end of the session, this is what we're going to look at. Phospholipids, you also have what we call steroids. Okay, so we are going to study all these. We're going to study the fats. I've not also included, you also have the oils, the waxes, fatty soluble vitamins, glycerides, phospholipids, and steroids. But one important thing that you need to understand about lipids is that they are water insoluble. They don't dissolve in water. And when you're talking about the property of dissolving in water, okay, the property of dissolving in water depends on the bonds. What type of bonds do you have in a compound? So if you have got a compound that has got a lot of carbon to hydrogen bonds, why am I talking about this? This is because these bonds are non-polar. Let me use non. So these bonds are non-polar. Because these bonds are non-polar, they are going to form non-polar molecules, and non-polar molecules cannot mix with polar molecules. That is why lipids are going to be non-polar. Why? Because they have got non-polar bonds, and that will make them to be insoluble. Remember the rule of like dissolves like. So like is going to dissolve like. So water is polar and lipids are non-polar, meaning these two guys cannot dissolve each other because like is supposed to dissolve like. And you need to explain why these are polar. It's because of the bond. Why they are insoluble is because of the bonds which are non-polar. Okay, that's very important. But lipids, since they are non-polar, you should look for a molecule that is also non-polar. That is why you can dissolve them. So lipids can be dissolved in non-polar molecules or organic solvents. And organic solvents will include things like ethers. That's where we can dissolve them. Ethers. You also have chloroform. Apart from chloroform, also benzene. So you can dissolve lipids in these liquids. Why? Because these liquids are organic. They are also going to have parts which are non-polar. That is very important. Now, let's talk about the functions. What are functions of lipids? Okay, what are the functions of lipids? Number one, if you know, be free to get to comment. So if your mic or your camera is on, kindly just switch them off. But if you want to answer, please be free to ask. Anyone who knows any function of lipid, any. Okay. One function of lipids. Yes, yes, Harry. Very good. They are going to be used in the storage of energy, just like carbohydrates and also proteins. Lipids are also used for energy storage. Actually, lipids will store energy more than anything else. And lipids can give us energy more than carbohydrates, more than also proteins. And the reason for that is that they are highly reduced. What do you mean by highly reduced? Remember, reduction by definition is how many reduction is the gain of hydrogen. So lipids have got a lot of hydrogens when you compare them to carbohydrates. So because of that, they can actually give you a lot of energy. You are going to understand this even more for some of you who will be at Ridgeway next year. Okay, and then they are also going to be used in what we call cell signaling. So how do cells communicate? How is one cell going to send information and communicate with another cell? That is what we call cell signaling. So lipids are also going to be important in that. Apart from that, they're also used in the cell membrane for structural support. Remember, the cell membrane is made up of a phospholipid bilayer, and that is a lipid. So they are used in the cell membrane. Apart from that, they are also used for heat and electrical insulation. Important is insulation. So think of a leaf, the way a leaf is. If you look at a leaf, there is that part of on top which is shiny, which we call the cuticle. So what happens is that what that is going to be able to preserve, okay, heat loss. That is the lipid that is found on that part. That prevents heat loss, like just like our bodies. Okay, the lipids that we have is going to prevent us from losing heat. They are there to act as insulators. Apart from that, lipids are also used in what we call buoyancy. What is buoyancy? Have you wondered why big animals like a whale, which is the biggest animal, can float in the water or on water? That is because it has got a lot of fat and that makes it be able to float on top. Lipids are also used for the production of metabolic water. Think of what are those animals which have got a hump on top of them? Those are camels, isn't it? So they can be used to produce metabolic water. So from the stored lipid, you can break down the lipids and then form water. That is metabolic water. Apart from that, you can also use lipids for protection of internal organs. For example, if you come in the abdomen, where our intestines, our stomach is, you are going to have a lot of lipid which will be able to act as shock absorber. Imagine something would easily just hit your stomach or hit your intestines. Some of these places which are inside, like the heart, are delicate, so they need to be protected, and what protects them is lipid. All right, so that is it on the functions. Now let's look at the different classifications and the types of lipids. We are going to start with the fatty acids. Trust me, if you are going to follow this one well, this class well, I'm going to make it short and to the point. You never miss not a single question on lipids. This one I can assure you, not a single question. As long as it is prepared at ONZA, unless otherwise. Okay. So fatty acids. Now what are fatty acids? Fatty acids are basically uh, carboxylic acids. But then these are going to have a long chain of hydrocarbon. So you are going to have their function, you are going to have an ara group. This ara group is going to be attached to a carbon, and then it's going to have a double bond with oxygen, and then it's going to have OH. So this function group that you can see, I can as well write it like this: ara bonded to carbon, bonded to two oxygens, and then an H. This function group you can see here is called the carboxylic acid functional group, or carboxyl. Please, this is not a carbonyl. We also have a carbonyl which is like this, COH. Carbonyl is the functional group for carbohydrates. But this one, carboxyl, is not in carbohydrates. C is the functional group for fatty acids. Okay. So the molecule for fatty acids, what you are going to have is that you are going to have a chain that is going to have a lot of. Okay, I've seen a hand. Yes, Dennis. Okay. So we are saying that carboxylic acids. Fatty acids have got a carboxyl functional group. And the carboxyl functional group is this one that you can see: carbon double bonded to oxygen and then bonded to OH. You can also write it as COOH. Different from carbonyl, which is a functional group found in lipids, I mean in carbohydrates. Now, fatty acids, you can have them like this. Look at this. You have got, I'm writing a carbon chain. This is carbon number 1, number 2, number 3, number 4, number 5, number 6, number 7. Let me put 8, 9, 10, 11, 12, 13, 14, 15, 16. So at the end, look at what we have. I can put at the beginning or at the end. You can see this functional group. This is the carboxylic acid functional group. Let me write another one. Carbon 1, 2, let me write it the way I've written the other one. Carbon 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18. I'm also going to put like that. But for this one, let me put a double bond somewhere. Okay. So 1, 2, 3, 4, 5. Let me have a double bond here. So I've got two fatty acids that you can see on the screen. Now for these fatty acids, I want you to determine we determine the formula. Each end that you can see is a carbon atom, each end. So we have got a carbon atom, and each end that you can see, as long as you have got a bond on each carbon, as long as you have got a bond on one side and on one side, that is two bonds. Carbon is supposed to have four bonds, meaning that this carbon has got two hydrogens. So this carbon has got two hydrogens, this one has got two hydrogens, this one also two hydrogens, two, 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 just like that. But then something that you can look, the carbon at the end, is only having one bond, so it needs three bonds. So this one is going to have three hydrogens. And then if you come on this one, still two carbons, I mean two hydrogens, two hydrogens, two hydrogens, two hydrogens, two hydrogens, two hydrogens. But look at here, you have got a double bond. So for this double bond, a double bond, you can see this carbon. How many bonds does it have? A single bond and then a double bond. Those are three bonds. So it only needs one hydrogen. Same one with this one, it only needs one hydrogen. But this one, two, two, and it continues. The last one will need the three. The fatty acid that you can see on top that doesn't have any double bond, we call this as a saturated fatty acid. It is saturated because it doesn't need any other bond. There is no place for the formation of another bond. The one which is down here is unsaturated. So unsaturated fatty acids, where you can see the double bond, you can actually add hydrogens there. But for unsaturated fatty acids, what you'll be able to see is not a way of written it straight because of the double bond which is there. Saturated fatty acids will end up making a kink. So instead of it going straight, it is going to go like this. So where there is a double bond, unsaturated fatty acids will turn, they form a kink. Take note of that for the sake of that exam that can come, right? So now let's determine the general or the chemical formula for these compounds that we can see here. Let's start with the saturated fatty acid. You can see at the end, we have got carbon, and that carbon, the functional group is COOH, like that. And then look at what is bonded to. It's bonded to a carbon with two hydrogens, right? As we said, carbon with two hydrogens. And you can see to continue another carbon with two hydrogens, another with two hydrogens, another. It will continue like that. So I can just put N here. And then at the end, the last carbon that you have has got three hydrogens. So this formula that you can see is the general formula for all saturated fatty acids. This is the general formula for all saturated fatty acids. I could as well written it by starting with CH3 and then I write CH2 with N and then I'm going to write COOH. This is very important, don't forget that. Because they can give you a formula, they'll just write the formula and then they ask you to determine whether it's saturated or unsaturated. So it should be able to balance with the formula that we have here. With the number of hydrogens, the number of carbons should be able to balance. And saturated fatty acids, you can see at the end, you also have CH3. And then the other carbons, you can see they are going to be CH, okay, CH what? CH2 with N. But are you able to see that after these CH2s, you are going to have one where you are going to have a double bond. So you are going to have a double bond somewhere. And then you are going to continue with your CH2 with your ends like that. But then at the point where I've got a double bond, you don't have two hydrogens for the carbon. You only have what? You only have one. So how are we going to write the formula? So this is going to be CH3 at the end, and then you have got CH, only CH, and then a double bond, another CH. Okay, because where the double bond is, you only have one hydrogen for each carbon. And then now you can have your CH with a 2, and then you can now put your N there. Remember at the end you have got the functional group which is COOH. So this is the general formula that all unsaturated fatty acids are going to be able to follow. But of specific importance, what I need you to understand, the unsaturated fatty acids can be mono unsaturated, di, or tri. Mono unsaturated just means that you only have one double bond. Di, two double bonds. And then tri, three, just like that. Okay. So let me first pause. Do you have any questions what I've talked about so far? Any question? Okay. The Saturated fatty acids, they don't have any double bond. And these saturated acids, because they don't have any double bond, there is no point where anything is supposed to come and there is no point where any hydrogen is supposed to come and add. These ones at room temperature, please take note, room temperature, that is like 25 degrees, they are going to be found as solids. Okay? They are going to be found as solids. And the most common example you find in exam is known as stearic acid. So what is stearic acid? Stearic acid is a saturated fatty acid that is having 18 carbons. At room temperature, you are going to see it as a solid. Now, unsaturated fatty acids, because they create this king, remember this double bond to make them to 10, right? To 10. So they are going to create that kink. So if you have got one fatty acid like this, where the double bond is, it tends. And then you have got another fatty acid like this, where the double bond is also tens. So are you able to notice that these two chains have gone away from each other? Because they have gone away from each other, that means that the forces of attraction between them are few. Because of that, they will not be solids. They'll end up being liquids. So at room temperature, unsaturated fatty acids are liquids. Take note of that, they are liquids. And they are going to be liquids at room temperature. And the very common example that you use here is something which is known as oleic acid. Oleic acid is unsaturated fatty acid with 18 carbons. Stearic acid is saturated fatty acid with 18 carbons. They both have 18 carbons, but then oleic acid is unsaturated, stearic acid is saturated. These examples you need to know, right? They are going to ask you. So where can you find some of this? So saturated fatty acids, these ones which you are going to find as solids at room temperature, you are going to find them in foods like butter, cheese, coconut oil, in meat, margarine, and also those foods that you fry, so fried foods. That's where you find saturated fatty acids. And then for the most common example I've already mentioned is stearic acid, which has got 18 carbons. Okay, so stearic acid is going to be a waxy solid at room temperature. It's a solid, like the way wax is. You know how wax is, isn't it? And then for the unsaturated fatty acids, they are going to have a double bond, at least one double bond. Okay, so because of the double bond, that makes them flexible, and that flexibility makes them to be fluid, and that's why they are going to also have a lower melting point. That's why they are found as liquids at room temperature. They've got that lower melting point. An example is oleic acid. Where are you going to find unsaturated fatty acids in what foods? Basically, think of lipids which form things which have got lipids which are liquid. So this will include olive oil, avocado, soya beans oil. Okay, basically oil, sunflower oil. That's where we're going to be able to find these ones. And then for the saturated fatty acids, these are solids, right? They are going to be used in the production of things like soap, detergents like washing paste, cosmetics like shampoo. You know, the shampoo, the shaving creams, that is the saturated fatty acid. Okay. And then on the same fatty acids, you are also going to have what we call the cis and trans fatty acids. So if you see, we are talking about this one, cis and trans only exists on the unsaturated fatty acids. So where you have got a double bond, right? You have got double bond, but then the chain moves like that and also moves like this. So the two groups that are attaching to the double bond are on the same side. That is what we call cis. But where I've got a double bond, one group is like that, another group is in the opposite direction. This is known as a trans. Okay. So this is what we call cis and trans fatty acids. Basically this. Uh, any question on fatty acids before we move from there? Any question? Any question? Uh, l- let's start with Moses and then we go to Peter. Yes, please. Okay, Peter. Okay, yes, go on. Okay, all right. So in a case like that, let's say they gave you a formula, CH3, and then they said CH2, and then they put here like a 7, and then they write COOH. You should be able to follow one of these formulas. You can see that this formula is agreeing with this one. Now, this doesn't mean that for the unsaturated, they're going to give you a double bond in the formula. As long as when you try, for example, they've given you that something is CH3, CH2, 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 and then at the end you see COOH. You just see the way the atoms, each carbon on the middle should have two hydrogens. So meaning that you are going to first, from whatsoever formula they'll give you, first remove one carbon with three hydrogens, and then remove a carbon with two oxygens and a hydrogen. The rest of the carbons that are going to remain, if you have got two carbons, look at this. For each one carbon is supposed to have two hydrogens. So if you have got two carbons, you need to make sure that you double the number of hydrogens. If it balances that, then that is saturated. For example, if I've got two carbons, I'm supposed to have how many hydrogens? Four hydrogens. As long as I remove the first and the last, if the carbons which have remained on the middle here are five, I'm supposed to have 10 hydrogens. As long as that balances, then it has proven with the formula for saturated. That means it's a saturated fatty acid. Is that okay? Okay. Yes, Peter. Okay. So when you see a fatty acid like this fatty acid which I've drawn on the screen here, the same oleic acid. So how are you going to remember that oleic acid is such unsaturated? Just think, for me, I always used to use oleic oil. Oil. I always know that oil is liquid. So oleic acid is liquid. Okay, what fatty acids are liquid and saturated? You remember like that. So if we have got something like this, you have got a double bond here, and then you have got something like this. Can you see this one? How about see that on the double bond, this group is down, this group is on top. So this is a trans. And then this group is down, this group is also down. So this one is cis. So that's what we mean by cis and trans. So because the groups are both down for a cis compound, this makes the compound to be more flexible than the trans. Okay, just know that this one is more flexible than the trans. Peter, is that okay? Okay. So that is it on the fatty acids. The next one, let's talk about glycerol as another lipid you need to know. So you need to know that all lipids that you find at room temperature as liquids, we call them oils. And all lipids that you find at room temperature as solids, we call them fats. So I've got fats and oils. Now glycerol, the other name for glycerol is known as glycerin. If you remember that, kama My bottle you used to drink when you are a child. Glycerin. So glycerol is a simple sugar polyalcohol. It is a sugar polyalcohol. What do you mean by polyalcohol? It has got a lot of alcohol functional groups. It has got a lot of OHs. Okay. So glycerol is going to be, it has got three carbons. The formula, I need to know the formula for glycerol. It is C3H8O3. Please, you need to differentiate it from trioses. C3H6O3. Remember for carbohydrates, carbon to hydrogen to oxygen ratio follows the ratio of 1 to 2 to 1. That is C3H6O3. But glycerol doesn't follow the ratio of 1 to 2 to 1. C3H8O3. So this formula is for carbohydrates. Trioses like glyceraldehyde and dihydroxyacetone. But C3H8O3 is for glycerol. And the way the structure looks like, you are going to have three carbons. You have got OH, OH, and the OH, and then the rest will be the hydrogens. Okay, this is glycerol. You can see the structure. Now, that's why we're calling it as a polyalcohol, because look at the OHs. I mean, are they? There are three of them. Okay. So what can happen is that, okay, I'm going to talk about that later on. So this glycerol one, it is that it is colorless. You just think of the bottle for glycerin that you use to that you put in your lotion. It is colorless, it doesn't have any smell, odorless, and it is viscous. Like when you pour it, it doesn't flow as fast as water flows. That means it is sticky. That's viscous. And it is also sweet. Just remember from everything about glycerin. We actually used to drink when we were children, that those things. Okay. And another thing is that because it has got these OH functional groups, these bonds are polar. So this makes this guy to also be what? To also be polar. All right. So it can dissolve in water. One thing I forgot about fatty acids. Fatty acids have had two ends. Did you see the way the structure was? It was like this. And then at the end, so at one end, it had the group which is polar. That means it is hydrophilic. This end, carbon to hydrogen bonds, these are non-polar, that's hydrophobic. A molecule that is both hydrophobic and hydrophilic is anyone? A molecule that is both hydrophobic and hydrophilic? Very good. Amphipathic. So if that comes in the exam, fatty acids are amphipathic. Okay, they have got one end that can be attracted to water, another and that cannot be attracted to water. So you can see we've talked about the characteristics of glycerol. Okay. Another thing that you need to know, what is the use? It can be used in the food industry as a solvent to dissolve things, also as a sweetener to sweeten things, also as a preservative. It can also be used in pharmaceutics, in medicines, and also in cosmetics, like in lotions, the making of lotions. All right. So that's a use of glycerol. Now, when we get glycerol, we combine with a fatty acid. Remember how a fatty acid is? You have got this group. So glycerol plus fatty acids forms a molecule that we call a glyceride. Please, this is not glycerin. Glyceride. This is C, this is E, glyceride. So what is a glyceride? A glyceride is a molecule that is formed from a combination of glycerin or glycerol with a fatty acid. So if you see a structure like this, so this is a glyceride. Now let's look at how this glyceride is really formed. So the formation of this glyceride is a dehydration reaction. Water is going to be lost. what is going to happen is that the OH coming from this guy is going to react with the hydrogen coming from this one, from the acid. And what you end up forming is going to be a structure like this. You can see the way we have combined it. So you are going to have this oxygen is still going to be there because this reaction will produce water. So that oxygen is going to be used to form what we call uh, a bridge, an oxygen bridge there. And then you have got this. And then the chain will continue like that. So if you have got glycerol and only one fatty acid, we call that as a monoglyceride. If we add two, like this is a diglyceride. If we have got three, like that, this is known as a triglyceride. The fatty acids that you are using can either be saturated or unsaturated, doesn't matter. But take note, a fatty acid plus glycerol, that gives you glyceride. Depending on how many fatty acids, because in multiple choice you are going to see some questions they say three glycerols plus one fatty acids is equal to triglyceride. Is that answer correct? Anyone to comment? Imagine you saw a question which said 3 glycerol plus 1 fatty acid is triglyceride. Is that correct or not? It is not correct. It's supposed to be 3 fatty acids, 1 glycerol. So same with the diglyceride, same with the monoglyceride. Please take note of that, alright? That's an exam question and an important one in that case. Do we have any question before we can move out of the or out of glycerol and glycerides? Any question? Any question? Okay. So no question, meaning that we have understood. Let's go to the next lipid, which we call a phospholipid. Now, a phospholipid was supposed to actually be a triglyceride. Okay? Actually, it was supposed to be a triglyceride. There is only one difference. Let me write a triglyceride. So here we are. We are supposed to have oxygen. You know how the bond is formed. And then another one. But just take note for this one. One chain is going to be unsaturated, another one is saturated. So that's why there's a kink there. But can you notice we don't have the third fatty acid? Instead, that fatty acid has been replaced by a phosphate group. Okay, it has been replaced by a phosphate group. So let me just put phosphate there, phosphate group. So this divides this molecule. So this front part here, we call it as the head. And then this end, we call it the tail. You can see the tails are two and it forms a kink, it bends. When we talk about cells, you are going to realize this is the structure that you find in the cell membrane. The head, because made up of a phosphate is negatively charged. So the head is polar, while the tail is non-polar. So in the cell membrane is a lipid bilayer, phospholipid bilayer. So what you are going to see is that you are going to have a chain like this. The circles I'm writing, those are the heads. So a cell membrane is like this. Think of the entire cell. The entire cell is all supposed to be like this. This is the cell membrane that I've drawn. This is the cell membrane. This is just one thing. Now it's bad that I've even shown two layers. There are no two layers, it's just one layer. That's the cell membrane. But the cell membrane, you are going to have the phospholipids. You can see the heads of the phospholipids can either face inside the cell or outside the cell. And then the tails are facing each other. That is because outside the cell you have got water. So you need the heads which are polar to interact with the water. Inside the cell you also have water. You also need the tail, the heads to interact with the water. But then the tails, remember, they are hydrophobic. So they don't interact with water. That's why they are hiding. And when we're talking about water and these properties, we talked about these, they can form what we call a missile. A missile is something formed like this. You can see like the heads will be together like this, and then the tails are going inside. They are hiding the heads are hiding the tail. So the heads can interact with water outside while the tails are hiding inside. This is what will happen when you add the phospholipid in water. It is going to form a missile. This is how it looks like. Okay, so that is about a phospholipid. Is there any question about phospholipids? We are good. Yes, Peter. I can't clearly get your. I don't know if you are using headsets. They are producing another sound. Uh-huh. Okay. Okay. So triglyceride, we are supposed to have a glycerol. This molecule that you can see on the middle here is glycerol. We're supposed to have three fatty acids. One, two. The third one was supposed to be attached here. But you can see we don't have the third fatty acid. Instead, it has been replaced by a molecule that contains a phosphate in it. So that third fatty acid has been replaced by a molecule that contains a phosphate. Is that okay? Okay. Another important thing is that phospholipids are also they are hydrophilic and hydrophobic. And don't forget, a molecule that is both hydrophilic and hydrophobic is known as amphipathic. Very good. Let's now talk about waxes. What is a wax? A wax is an ester or fatty acid with alcohol. So when you combine a fatty acid, okay, fatty acid plus alcohol, that is what will form what we call a wax. And this thing here, trust me, everything. I'm actually, what doesn't come, I'm not talking about it. Okay. So what you are going to see is, let's draw a fatty acid. This is a fatty acid. And then at the end, you know the functional group for a fatty acid. And then an alcohol, the functional group is OH. And then it's like that. Because what they'll do is that they'll just give you the structure. They ask you to determine what you have. So what you are going to see is that these are going to react like this. The hydrogen from the fatty acid will react with the OH from water. And so the bond that you are going to see on the middle, you are going to have double bond with oxygen. Remember, there is also this oxygen that hasn't reacted. And then you are going to have the the alcohol this side, and then you have got your fatty acid this side. So the side where you just see the double bond with oxygen, that's where the fatty acid is coming from. So this is what we call a wax. This is what we call a wax. It's an ester. Okay, it's an ester. It's going to be found. It's going to be used for waterproof. You know how waxes are. I think who has never seen wax before? Even in our ears, sometimes we remove wax from our ears. You know how it is. So it is going to be used as waterproof. And in plants, they can form a protective layer for the leaf. You know how a leaf of a plant is? Just a mango tree, if you get a leaf from there, the outer part is a bit shiny. That is known as a cuticle of a leaf. And that is going to be necessary in preventing water loss. Okay. And then in animals with a backbone, vertebrates, they are going to be found in the skin, in fur also, I don't know how you pronounce that, of fur. They're going to be found also there, and also in feathers and in insects. You know, insects have got the skeleton that is outside. We call it as the exoskeleton, right? So all this, and that is going to be all used to protect the animal, all right? Like for example, in the skin, avoiding water losses. We have got what we call beeswax. Beeswax is a special type of wax which is going to be found in the honeycomb. You know the honeycomb, that thing that we eat. Okay, the honeycomb. And what you are going to, that is where it's found. So the bees use it to store honey and also to protect the larvae and the pupa. The larvae and the pupa, these are just stages of the bee as they get to grow before you can have the actual bee. And humans will use it in the food industry as a glazing agent. Okay. And what else? You can as well use them. They are also used in cheese, chewing gums. They are also all going to be found in those things. Okay. So basically that works, especially on the structure. If you see it, make sure that you can tell. Another lipid you need to know is what we call steroids. What are steroids? Now, steroids are special lipid compounds which have got four rings. These ones have got four rings. Okay. And of these rings, you are going to see some of them, they are going to have five carbons, others are going to have six carbons. So we're going to see something like this. So these are two rings, and then you see something like that. How many carbons are these? Okay, let me draw this well. You can see these three that I've drawn, they have got six carbons each. And then one is going to have five carbons. It's going to appear like this. So this is what we call a steroid. And then at the end, normally you are going to have an alkyl functional group, OH. Okay. So these same steroids that you can see, the rings that you can see like this, they have got a lot of carbons and hydrogens because each end that you can see has got carbon and hydrogen. So this makes the steroids to be non-polar, and the most common type of steroid that we talk about is cholesterol. Cholesterol is very important and is going to be found in animal cell membrane. What is the function of cholesterol? Cholesterol is going to be used as a precursor of fatty soluble vitamins. Remember the fatty soluble vitamins, ADEC. Precursor is just something from where something else comes from. So the vitamin A, vitamin D, vitamin E, vitamin K can actually come from cholesterol. And you also have what we call steroid hormones. Steroid hormones. Okay. Hormones like testosterone. These hormones are going to come from cholesterol. Very important. You've heard of hormones like estrogen, which control the characteristics of a female, the secondary physical characteristics like having breasts and other things like that. These are steroid hormones, okay? Basically, sex hormones. So these are lipids. We have done the functions of lipids. We have described the different types of lipids, and lipids is done. The entire topic is done. Let's look at some questions. I just included a little questions from exam question. Let's start with this one. Steroids hormones are made from amino acids, cholesterol, triglycerides, waxes, fatty acids. I don't know. Which one would you pick? Cholesterol. Okay. Wilfred, your hand is up. You had the question? Yeah. Okay. Anastasia, your hand is up. Anyone else with a different answer or anyone to agree if cholesterol is going to be the answer here? Okay. No one wants to speak. Question 2. When a lipid is mixed with water, the two immediately separate because the lipid is amphipathic, the lipid is denser than water, hydrogen bonds form between water molecules, the lipid is hydrophilic, hydrogen bonds form between water molecules and lipid. Which one do you think is the answer here? When lipid is mixed with water, why do the two immediately separate? Is it that the lipid is amphipathic? The lipid is denser than water. Hydrogen bonds form between water molecules. The lipid is hydrophilic. Hydrogen bonds form between water molecules and the lipid. Which one would you choose? For the first one, okay. Okay. Anyone else? Guys, let's speak. You don't want to First one. Okay, so this is the answer. The best, if it was hydrophobic was there, I would have picked hydrophobic. But amphipathic covers both hydrophobic and hydrophilic, so that would be the best. Identify the lipid that has got charged component. Charged component. Phospholipid, triglyceride, wax, cholesterol, glycerol. Which one would you pick? Okay, Harry picks one. Anyone else? Wope also picks one. Anyone else? Gift picks one. Anyone else? Okay, Anastasia goes for two. Anyone else? Okay, remember for phospholipids add a phosphate, and I said that the phosphate head is negatively charged. And triglycerides, they are going to be the triglycerides. When you remove one fatty acid, that's when you can form a phospholipid. So triglycerides don't have a charged component. Okay, so the best would be one. Let's look at this one. An ester bond is a link between an OH group of carboxylic acid and OH group of alcohol, C double bond to oxygen group of carboxylic acid and an alcohol of a carbon, and an OH group of fatty acid and CO group of lipid, carbon of fatty acid and OH of a lipid, OH of a glycerol and OH of another alcohol. Which one would you pick? Okay, one, one. Okay, anyone else? Anyone else? Another option? Okay. So can someone explain for me why are you picking one? Those who have picked one? Okay. An OH group of carboxylic acid will react with OH group of an alcohol. Okay. Why is option 2 not correct? An alcohol of a carbon, right? And okay, good. And then option three, an OH group of fatty acid and CO of a lipid. Why is that correct? Why is that not correct? Okay, but remember the double bond with oxygen is found in all lipids. At the end, what determines saturated and unsaturated is the end here and not this group here. Okay, the wrong thing here is that this question is not this option is not specific. Look, OH group of a fatty acid and CO group of a lipid. This CO group is not supposed to be of a lipid, it's supposed to be of an alcohol. Okay, and then for option 4, a carbon of a carbon of a fatty acid and OH of a lipid. That is not even a carbon. OH of glycerol and OH of another alcohol. OH of glycerol and OH of another alcohol. And you know that glycerol is not supposed to be with an alcohol, it's supposed to be with a fatty acid. Unless if it was fatty acid there, the option would have been correct. Let's look at this one. Identify the molecules which are highly reduced form of carbon. Monosaccharide, disaccharide, polysaccharide, triglyceride, waxes. Identify the molecules which are highly reduced form of carbon. Disaccharide for one, monosaccharide, they contain carb. Okay, I know it's just English which is confusing you here. Identify the molecules which are highly reduced form of carbon. If we say highly reduced form of carbon, we just mean which one of these molecules here have got a lot of carbon to hydrogen bond. A lot. Which ones amongst these have got a lot of carbon to hydrogen bonds? Obviously not monosaccharide because monosaccharide has got fewer bonds than disaccharides. Okay. Triglycerides have got a lot of carbon to hydrogen bonds. Same with waxes, same with polysaccharides. But polysaccharides don't end. Like the molecule continue going on and on and on and on and on. So that would even be the best because we don't have a specific number of chains that can come at the end. Triglycerides, which are made up of highly reduced, meaning you have got a lot of hydrogens attached to a carbon. So you are looking at molecules which have got the highest number of hydrogens. Which molecules have got the highest number of hydrogens? Okay. Triglycerides, which are made up of a dash, are liquid at room temperature. Glycerol only, cholesterol, and saturated hydrocarbon chain, saturated fats. Which one would you pick? Remember, are liquid at room temperature. Unsaturated. Okay. Another one says unsaturated. Anyone else? Unsaturated. Okay, very good. That's the correct answer. Lipids are partially soluble in water because they are amphoteric and neutral, have hydrophilic portions. They do not have a charge on them, have hydrophobic tail. Which one would you pick? Okay. They have got okay. So we have got two options. Let me hear from others. Which one? Three. Three. Okay, good. So we are picking three because the question is saying lipids are partially soluble. What explains why they are soluble is hydrophilic. But if the question why are they insoluble, we would have picked five as the answer. All right. And then let's answer this one. Lipids are esters of fatty acids, fats and oils, polymers of fat acids and glycerol, esters of fatty acids and alcohol, not the same as triglycerides, polymers of phospholipids. Which one would you pick? What are lipids? The first thing we talked about. Okay, Ali goes for three. Anyone else? Anyone? Okay, Anastasia goes for two. Okay, Gift goes for three. Anyone else? Arnold, what are you saying? Okay, okay, Arnold goes for two. Okay, Wope goes for one. This one is nice, huh? Okay. So this was the introduction part. Look at this. Look at what I said about lipids. It's here. It's here. It's here. What are lipids? Are you able to see the answer? So, since you have seen it, so which one is the correct answer? Okay, so 3 is the correct answer. Okay, and then here they've given us C3H8O3. C3H8O3 is a fatty acid, a lipid, an alcohol, a steroid, a triose. I don't know. Which one would you pick? Okay, a trios, and then Ari says one. Anyone else? Anyone else? Okay, it will be one. Okay, Wilfred, say lipid. Okay, remember a trios is supposed to follow the formula C1H2O1. So it is C3H6O3. So this one they've given here. What is the specific name of the lipid? It's glycerol. Very good. Glycerol. The following substances are lipid in nature except, so the formula for glycerol is similar to the formula for trioses. Okay. So trioses have got the formula C3H6O3. The ratio of carbon to hydrogen to oxygen is supposed to be in ratio of 1 to 2 to 1. But you can see this formula, if you divide through by 3, you not get a ratio of 1 to 2 to 1. So that meaning that's not a carbohydrate. But glycerol, which is a lipid. And for you guys who have picked one is not correct. Option one is not correct. I thought it was the answer is a lipid. Okay, it's a lipid because all these glycerols, these are all lipids. Everything that we talked about are lipids. A fatty acid doesn't follow this formula. This lipid is called glycerol. Does that make sense? Does it make sense? Very good. The following substances are lipid in nature except glyceride, wax, cholesterol, fatty acid, insulin. Which one would you pick? Okay, someone at five, another one. Any other one? Same five. Okay, it's insulin. Very good. And then the last question. The hydrolysis of lipids produces glucose, glycerol, fatty acid, amino acid 2 and 3. Which one would you pick? The hydrolysis of lipids will produce what? Okay, option 5. Anyone else? Okay, very good. So the answer is option 5. Just in case you are lost, just in case you are lost, look at this. Work says we were forming them by removing water. Okay, removing water. So to a wax, if you add water, you are going to break it. That is what is called hydrolysis. So you can form a fatty acid and an alcohol when you add water to a wax, because when we are forming it, we were removing water. Another one, a phospholipid. A phospholipid, we are getting glycerol, we react with the fatty acids, and we were removing water. So if you add water back to the molecule, you are going to break it down. That is known as hydrolysis. Okay, same with the glycerin or glycerides. When you add water to them, you are going to break it into glycerol and fatty acids, because when you are forming them, you are losing water. That is why for this one, breaking of lipids, you can get glycerol, you can as well get fatty acids. So that's why the answer is option 2 and 3. Does that make sense? Okay, very good. So if you have enjoyed and you feel nice, you want to continue with your friends who are learning, you can get to register with us. You can inbox me on WhatsApp to get registered in math, math, biology, chemistry, and physics. And here is where the class ends. The next thing that we are doing in biology is going to be nucleic acids. I think that will be the last topic. Then we are done with the first semester biology. And then we'll just be revising test one papers, because we are done with all the topics for test one. There are very few. Actually, we also do cells, but normally they don't include cells in test one. But sometimes if they finish, they're supposed to include it in test one. For those people who have got biology 1401, just after doing cells, you're supposed to write the first exam, the first final exam, and then you write a second final exam, which will be the second part of biology. Otherwise, that is it. Thank you so much. Enjoy your evening.